Hello, dear friends, and welcome to IVMED webinars. This is the series of educational webinars held by fertility specialists of Medical Center IVMED and specialists of IVMED Family IVF and Surrogacy Agency. We are located in Kiev. My name is Nadia Milinevska. I am head of international department and I am a director of IVMED Family Agency. Today we have a very special webinar, and I would like to introduce the topic of this webinar. I just want to remind you that we have active chat. You can write your questions, and after presentation of our speaker, you can ask any question to different specialists and receive the answer. The topic of webinar is embryology laboratory, standards of cryostorage, work arrangement at Medical Center IVMED. Today we will discuss, according to multiple requests from our patients, the next issues. What is a cryobank? How the cryostorage of embryos and other biomaterial is organized at IVMED. IVF ID witness system for identification of patient and his or her biomaterial. Biomaterial collection and cryopreservation. The control of cryostorage safety at IVMED. And I would like to introduce our main speaker, Birol Aydin, Director of IVF Laboratory at Medical Center IVMED, Senior Embryologist, Official Expert of Society for Human Reproduction and Embryology. Medical Center IVMED is proud to have him in our team. We have today a very special way of representing our webinar, and today the whole embryology team is ready to give also answers for your questions. And we would like to introduce you also fertility specialist Veronika Ulanova. She's a medical doctor, obstetrician gynecologist, fertility specialist, doctor of the highest category. She is co-founder of Medical Center IVMED. So after presentation, you will be able to ask you any question from any area to our specialist. And now let's start. Birol, please.
Hi everyone, welcome to IVMED Embryology Laboratory. Today we will try different concept of webinar. So we are directly connected from our laboratory. You will be able to see our system, advanced technologies, and you will meet with our teams for your question and answer. First of all, I want to give information, technical information about IVMED Clinic Embryology Laboratory. So how our laboratory is working. Actually, we try to manage our laboratory with advanced technology for the special air quality standards, special equipment quality, and also for security systems of the laboratory. I will try to introduce our laboratory. So we are basically using two microstation which we are able to do fertilization, assisted hatching by the laser system, embryo determination, and also embryo culture follow-up during the process for the quality and the assessment. Also, we have special benchtop incubators which give us possibility to follow your embryos and biological material during the cultivation. So difference of the technology, it's allow only one patient in one well. So when we open the lab, so we don't give damage to quality of culture to other embryos or other patients. Also, we use special laminar cabin. So it is already 100% under sterilization condition. There is no particles, there is no any kind of bacteria, infection, or pathogens which can survive inside. So all manipulation for the embryos and oocyte culture is doing under laminar flow coverage. So we are protecting all this system. As you see, many different tablets on the wall. So all of them is making quality management for the incubators, for the laboratory, and for the air quality. We can be able to measure 24 hours pH standards in the culture dish, how is developing. We can measure for each well temperature also. Also, we can measure different particle levels, pressure, and humidity. And we are able, even from our home, from our phone application, to follow the patient which we have in incubator. Also, we have time-lapse technology, which help us 24 hours and 6 days of culture to follow up as the time-lapse dynamic video of the embryo and oocyte. So while we have your biological material as oocyte and embryo inside of time-lapse, we are able already to see how embryo developing. So when embryo start to get fertilized and to get 27 hours, it gives us big potential for the implantation of embryos. We choose high quality of embryos by the time-lapse. Time-lapse gives us main point which embryo is the able and more, it can give more chance for the pregnancy and also implantation. So now I will try to give some tricky information about our biological safety witness system. So we create for you a scenario, a patient, how we are going from the function till embryo transfer with witness system. Now we will visit our operation room and we will try to see how we proceed this system. So as you see from each panels, we have special card reader, which is connected to the main screen. When we have the patient or surrogate for the embryo transfer, each surrogate or patient has side bracelet, and when we read this smart chip, which you see inside, already we found information of patient, donor or surrogate, and also dynamic picture of the embryos for the embryo transfer. So when we go to, for the embryo transfer to our embryologist, so our embryologist will show how system is going on. So special chip reader can read the chip, which is your culture dish for the embryo transfer, and she can determine the real patient. If she read by the mistake a different patient, system will not allow to make embryo transfer. Now she will try to read the wrong chip, and system will not give permission to do embryo transfer. So when she tried to read the right chip, then system proceed the embryo transfer, and patient can follow this embryo transfer on the main screen. You can see about embryo picture and the quality of the embryo transfer. While the same situation it happens for the function. So if we do a function for the donor programs, when donor come inside of our operation room, so when we read the chip, all donor information is coming directly to our screen. So name of doctor, possible number of follicles, and all information about donor can be seen on the main screen. Then when we move the embryologist proceed oocyte pickup process by the witness technology. So each step, she reads smart chip and how many eggs she found, patient and also doctors can see real time on the main screen. When function ended, she already made a culture chip in each petri dish. So for the witness process, each culture dish includes a smart chip. So in this case, there is no chance to mix up any kind of biological material. So when she signed the petri dish and the put chip, Then we are going for next process to our embryology room for humorous complex cleaning, as we are calling denudation. Our embryologist start to make the denudation process. So she will chip head on the culture dish, and she proceeds denudation. When denudation complete, we are already ready for the ICSI fertilization process. She will complete the denudation. Denudation complete. After the denudation complete, we will test the ICSI process. So now we are going to test ICSI process. So in the same station will be in the ICSI process. So each process, we will read the smart chip by the each culture dish, and each embryologist for the special process will proceed the system. So when we come here, system will ask also semen sample to read, and then we will proceed all system to finish with the culture dish. So if we are doing same things for the cryopreservation, so now we will visit our vitrification cryopreservation room. So we have also for each cryo special RFID chip, which follow up, which include all information of patient. When I read the special chip, so when I see the cryo, I already will see the potential of the embryos. So when I read the chip, already I will prove the name of patient, and there will not be chance for the mix-up of the materials. So as you see, all the process in embryology laboratory, we are trying to do under some security programs. So there is no risk to mix up of the material. There is no risk any kind of culture problems during the process. Actually, as a patient, you are only choosing the clinic, so you don't know the clinic behind what kind of laboratory they have. Most of the clinic has very beautiful buildings, but they don't have enough invest in the laboratory, enough technology in the laboratory. That's why we try to share with you under transparency all condition of our laboratory, how advanced technology we have, how quality management we have. So now we are ready for your questions, and we will be glad for each embryologist in our team give answer to you. So dear Nadia, we can pass the questions.
Thank you. We have questions. Hello, thank you for the webinar. I have already 5 IVF trials before, and I never had blastocyst for embryo transfer. What can be the reason for that? Do I have any advanced technique which will help me? Do you have, maybe?
Actually, when we have problem on the embryo stop on the step, especially on the cleavage stage or 5-day of blastocyst stage, of course, that can be a lot of different factors. So genetical factors, culture factors, laboratory factors, also, of course, embryologist factors. So in this case, it is difficult to determine this question by the genetic test, because there is no blastocyst which we can take biopsy material. But of course, with special culture techniques, we can use advanced technology of the time lapse. We can determine why this embryo stop and which stage stop. We can check sperm sample male factor with sperm fish testing. So there are different kind of way how we can find out why this embryo is not developing. But still, if there is question about developing, we have to advise to our patient that there are 2 ways. First way is egg donation process, and second way, if she doesn't want to do egg donation, mitochondrial spindle transfer or chronicler transfer.
Thank you. How you do oocyte pickup and embryo transfer safety during COVID-19?
I can answer this question because actually our embryological laboratory is the safest place, I think, in Ukraine now for sure. Because even usually, not during pandemic, we always do all disinfection after every case. Like all surfaces are cleaning with embryo and the oocyte safe disinfectants. Also, we working just in the safe laminar box. They have special airflow that prohibit contamination with any bacteria or viruses inside our culture dish and like biological material. We always work just with sterilized plastic, one-use plastic, like always really safe. During pandemic, we restrict the number of cases, so it was not so crowded and we do not have like too much patients. So we are ready for any troubles and like pandemic situation.
Thank you. I also have a question to doctor. A good embryo, 5AA, was transferred to me, but it did not implant. What could be the problem? Nadia, translator, please translate question, doctor.
Секунду. У меня был перенос эмбриона, хорошего эмбриона 5АА, но он не имплантировался. В чем может быть проблема?
Здравствуйте. Может быть несколько проблем, которые привели к тому, что отсутствовала имплантация при переносе хорошего эмбриона.
There actually might be several reasons that could influence the failure of implantation when the good embryo was transferred.
Первая проблема возможная – это изменения в иммунном статусе.
The first possible reason is the changes in the immunological state of the person.
Для того, чтобы понять, нет ли этой причины, надо обследоваться на предмет наличия антифосфолипидных антител.
In order to exclude this reason for infertility, you should be investigated for the presence of the antiphospholipid antibodies.
И повышение активности натуральных киллеров.
And the increased activity of the natural killers.
При наличии антифосфолипидных антител проблема заключается в том, что в итоге это реализуется микротромбозом, в том числе и сосудов эндометрия, в результате чего в него хуже происходит имплантация. Это снижает вероятность наступления беременности даже при переносе хорошего эмбриона.
In the case of antiphospholipid antibodies, the reason for infertility can be the microthrombosis, which decreases the probability of occurrence of the pregnancy.
Если повышена активность натуральных киллеров, то есть макрофагов, клеток иммунной системы,
if we are considering if the reason is the activity of the natural killers, macrophages,
это приводит к тому, что они считывают плодное яйцо как чужеродное, также препятствуя его имплантации.
They believe that the fruitful egg is the outside body, so they prevent it from the implantation.
Наличие такой проблемы требует параллельной в протоколе ЭКО иммунологической коррекции, что повысит вероятность наступления беременности.
If you have such a problem, then the most reasonable solution would be to run the IVF procedure along with the immunological treatment. Then the chances would be higher getting.
Вторая возможная причина – это изменения в эндометрии.
The second possible reason are the changes in the endometrium itself,
которые могут приводить к снижению его рецептивности, способности понять, что это плодное яйцо, его захватывать и втаскивать внутрь себя.
These can lead to the decrease in the receptivity of the endometrium, so the endometrium doesn't recognize the fruitful egg, and it doesn't attract the fruitful egg to itself, and it prevents implantation.
Для того, чтобы исключить эту проблему, нужно сделать такое исследование, которое называется иммуногистохимия эндометрия.
To exclude this reason, you need to do a special examination, which is called Immunohistochemical Examination.
Это исследование позволяет исключить такой статус эндометрия, который называется метэндометрит, который, в свою очередь, приводит к снижению этой самой рецептивности.
In this case, we can exclude the condition of metaendometritis, which will exclude one of the reasons for your possible reasons of infertility.
Если выявлена эта причина, это требует терапии, терапия противовоспалительная назначается и контрольной иммуногистохимии, и при пролеченном метэндометрите опять-таки повышается вероятность наступления беременности.
If this is a reason, it requires additional therapy, and at the end of this therapy, this stage should be checked again by the method of chemical hysteroscopy.
Также приводить к тому, что отсутствует имплантация, могут определенные изменения системы свертывания крови.
Another reason for infertility could be the changes in the coagulation system.
Для того, чтобы их исключить, делается исследование, которое называется гемостазиограмма.
In this case, you should do the hemostasiagram to make sure that this is not the reason why you're infertile.
Если выявлены изменения в системе свертывания крови, они подлежат коррекции, и при параллельной коррекции также повышается вероятность наступления беременности.
If this is the reason for infertility, then this situation can be helped by a treatment, and it should be performed along with the IVF attempt.
Вот это те причины, которые могут приводить к отсутствию имплантации хорошего качества эмбриона.
These are the reasons that may prevent the implantation of the embryo of a good quality.
Thank you. And it seems we have a question to embryological team. Why do embryos stop growing after day 3? Sperm and or egg factor?
Hello, my name is Olga, and I will try to answer it in simple words. To be fair, both eggs and sperm may be the reason why embryos stop their development after day 3 of culture. And the major problem in this situation is chromosomal abnormalities. Usually oocytes more often possess chromosomal abnormalities, but at the same time, the spermatozoa is one who actually controls the dividing of chromosomes in embryo. So both of them may be the fault when we talk about genetics. And also the additional cause, the energetic cause, well, unfortunately goes only to oocytes, because mitochondria, the energetic system of the cells of embryo, they are actually got from oocytes. So unfortunately, we are not able to say who is the fault in each particular case, but still can find ways to compensate this problem anyway. Thank you.
Thank you. If I have low quality of eggs still, can I freeze them? Maybe also to embryologist.
Okay, everybody, hi. My name is Maxime. I'm a embryologist in IVMED clinic. Okay, if you have low quality of eggs, so embryologists can choose like two options, or it's freeze or not to freeze. If you choose freeze this eggs, so this embryology must have good experience and good media. In IVMED clinic, we have the both. If embryologists choose do not freeze, so he thinks these eggs will not survive after thawing. If you have like this situation, you can ask your doctor or directly embryologist, and they can in details explain why they choose like this option.
Thank you. I had 3 IVF cycles with 3rd day embryo transfer. What do you prefer for embryo transfer, 3rd day or 5th day? Who wants, maybe doctor or?
Basically,
embryologist.
yes, basically as embryologists, we always prefer day 5 transfers because this helps us to understand whether embryos of this particular patient are able to reach the blastocyst stage that is very important for implantation. Because only blastocysts can go into uterus and give a pregnancy. So if embryo stops on day 3, well, we have no chance to get pregnancy at all. That's why we prefer to have prolonged culture of embryos, to be sure that they have high implantation chances, to be sure that they form blastocysts. We also can perform genetic testing at this stage, at blastocyst stage. That's why it's always better to go for blastocysts. But if you have only one embryo, for example, or 2 embryos, and we are not able to select out of them because there's only 1 or 2, we can go for day 3 transfers. But of course, if you already had such an experience, it is better to switch to blastocyst culture now and to look whether your embryos are able to go for blastocysts at final stages. Thank you.
Thank you. Hello, I have very low AMH, and finally I can have only 1 or 2 oocytes. Do you have some advanced technique which will increase my chance for fertilization and high quality of embryos?
So I think I can give answer to this question. When, of course, anti-Mendelian level is low, so mainly we get limited number of oocytes, and mostly quality of oocyte is low. So what we are doing in this case, of course, our main priority to try to get best embryo with such condition. Yeah, sometimes it's surprising. Yeah, even with low quality of eggs, we can get better quality of embryos while we are using different sperm chip technology, time-lapse technology, which we are giving less stress to oocyte and culture during the process. But mainly, of course, when anti-Müllerian is very low, and when we have the problem, the maturity, also we have advanced technology of in vitro maturation. So when oocyte is not mature because of small follicles and low anti-Müllerian level, with additional technique, with in vitro maturation technique, we can stimulate oocyte under laboratory condition, and we can maturate the oocyte. Then we will have more chance for the potential embryos. When we don't have such chance to get better quality of oocytes, there is only 2 possibilities. As first is like egg donation, and second, mitochondrial chronicler transfer.
Thank you. We have a question for doctor. I am 28 years old. I do not plan to have children yet, but the doctor says that I have low AMH, 1.2. Is there a reason to get worried and to turn to IVF in the future?
Вопрос врачу. Мне 28 лет, мне пока нет в планах заводить детей, но доктор говорит, что у меня низкий АМГ 1,2. Есть ли причина беспокоиться и обращаться, прибегать к ЭКО в будущем?
Действительно, цифра 1,2 антимюллеровского гормона, уровень, говорит о том, что это низковатая для 28 лет цифра.
It is true that for 28 years old, the AMH of 1.2 is a bit low.
Цифра это говорит еще о норморезерве, то есть о достаточном еще количестве яйцеклеток в запасе на сегодняшний день. Но эта цифра прогрессивно будет снижаться, она снижается у каждой из женщин, и в 28 лет 1,2 говорит о том, что времени для реализации репродуктивных планов, в общем-то, не так много, поэтому имеет смысл об этом задуматься.
So in this case, the result of 1.2 of AMH says that for now, the ovarian reserve is fine. But this result is quite low for your age, which means that it will only decrease with time. which means that now you're still in time to do something about it, but you have to think about the future and you might have not so much time to realize your reproductive plans.
Что может позволить сохранить репродуктивный потенциал? Это заморозка ооцитов и их хранение для того, чтобы, когда возникнут репродуктивные планы, можно было их использовать и получить беременность.
The best way to preserve your reproductive potential would be to freeze your oocytes now, so that when you have a desire to have a baby, you will be able to have your own cells to in cryo.
При антимюллеровском гормоне 1 и 2 можно сделать стандартную стимуляцию, она позволит получить достаточное количество клеток. В возрасте 28 лет они будут хорошего качества, что позволит их потом использовать активно.
With the present level of AMH, it's possible to run an IVF cycle with a normal protocol, and at this age, the majority of the cells will be of good quality. So you'll have no problem to freeze the biomaterial for the future use.
Если не реализовать идею заморозки ооцитов, тогда нужно мониторировать уровень антимюллеровского гормона, и если он будет снижаться достаточно быстро, все-таки рассмотреть вариант заморозки ооцитов. Спасибо.
If you're not ready to freeze your oocytes right away, the good idea would be to monitor the level of the AMH. So in case you see that it decreases rapidly, then it would be still a good idea to freeze your oocytes for the future use. Thank you.
Thank you. Will I have opportunity to see embryos before embryo transfer or during process? Embryology team.
Hello, my name is Elena. Yes, of course, you have opportunity. Before embryo transfer, we again check embryo quality and make photo. After we put a picture in computer in a patient card. And after coming in operation room, patient can see on the screen all information about yourself and can see embryo, how quality we make a transfer in uterus.
Thank you. How critical, can we improve the chance of success by transferring fresh or frozen embryos?
I think better make embryo transfer frozen embryos because we make perfectly prepare endometrium for embryo transfer.
Thank you. How critical do you feel embryology laboratory air quality is affecting embryo quality?
So, of course, quality of laboratory, as we talked when I present about laboratory, is one of the key points about culture condition and embryo developing. So, mostly, of course, we are investing a lot for the air quality. We are using special filter systems, HEPA filter systems, which is protecting about particularization and pathogens. We are using carbon filter systems, which is protecting our laboratory from kind of smells and chemical toxins. So all of this give us big opportunity to protect our embryos from the contamination. Of course, that is not installed for every laboratory because this is advanced technology. It is cost a lot, invest a lot, but as IVMED clinic, we are trying to give best condition to our patients, and our laboratory installed high quality of air quality system, and we are avoid all kind of particles and pathogens during our culture and culture system and embryo development. Thank you.
Thank you. We want to start IVF treatment again, but my husband will not have chance to travel and visit your clinic day of egg collection, on a day of egg collection. Can he freeze a sperm sample before egg collection? If he freezes sperm, that will that will affect the quality?
Hello, thank you for the question. Of course, he can freeze his sperm before all manipulations. Nowadays, vitrification is advanced technology which helps patients who is not able to come at the day of IVF treatment. Also, we recommend to freeze the sperm or if patients will have some long treatment, for example, chemotherapy, which give harmful influence at sperm. And in this situation, cryopreservation is the best way for saving sperm with good quality before some medication. So of course, as the case of your situation, cryopreservation of sperm is good variant for you. By the special technology of vitrification, spermatozoa will have high safety because we use modern cryomedium for vitrification, and this cryomedium keeps the function of sperm cells for a very long time. And also material will be stored at the right temperature of nitrogen, and we can use material when we need to. But of course, part of spermatozoa are losing their motility after thawing, but it doesn't affect on quality of ICSI procedure. Thank you.
Thank you. I have several IVF trials, but we could not get any success. My husband had bad quality of sperm morphology. Do you have any special technique which you can solve our problem?
Yes, we have special technique. One technique names swim up, and the second one is based on centrifugation of sperm with a special gradients. We use swim up when our sample of sperm has good indicators. The essence of the method is to do centrifugation of sperm with the culture medium, and after centrifugation, we use this culture with the sperm for insemination and another IVF treatment. And when we have bad quality of sperm, for example, bad motility, bad concentration, or bad morphology, like in your situation, we use special gradients. And using their gradients, only spermatozoa with the best motility and good morphology can pass through this gradient, forming the precipitate at the bottom of the tube. We will get portion of sample with high percentage of spermatozoa with good morphology and good motility. And after that, andrologist gives this sample of this sperm to the embryologist. And among these spermatozoa, they choose the best for making ICSI and other IVF methods. So yes, we can solve your problem very easy. So don't worry, we can help you. Thank you.
Thank you. Hello, when we have teratozospermia with 2% of normal sperm, is the fertilization rate with ICSI good?
Hello, I will answer this question. Yes, in our lab, we have a special technique called as microfluidic sperm sorting tip. I will tell a little bit more about this technique because it's suitable for all cases with high DNA level and also for bad morphology. It's chemical-free method. It's based on natural sperm selection in a passage through microbarriers, imitating natural environment of female reproductive system. Selected sperms have better morphology and genetic quality and motility. So that's why it's recommended not only for bad sperm quality with bad morphology and also for clients, for example, with low rate of fertilization with ICSI or poor quality embryo in the previous IVF cases. So yes, it's possible for us to work with such a morphology, and the rate of fertilization is probably is going to be good. Thank you.
Thank you. I have many IVF trials and egg donation cycle already, but we could not get pregnancy. I've heard PGD or NGS can help. Can you give me information? Do you have such technology? Do you have such technology in your laboratory?
Yes, we work with genetic testing of embryos. We can perform either testing for major chromosomal abnormalities with PGT for 5 or 9 chromosomes, or comprehensive chromosomal screening, so-called NGS technique, that will give us full information about embryo chromosomal health. So it may help to select the most healthy embryo according to its genetics for the subsequent transfer, and avoid unsuccessful infertility treatments. So will come if you look for NGSP or PGT 549, you can actually work with our IVF laboratory. Thank you.
Thank you. How you avoid human factor risk for oocyte and embryo mixed up in your laboratory?
Okay, I can answer this question because as you saw already, we are using IVF witness system for tracking all our dishes as biological material. We put chips on sperm tubes, we put chips on our culture dish, and also with our IVF witness system, we also use like a second witness, because I cannot change the culture dish without second embryologist who will watch me. So we use like double safety protocols. We use witness system and we use second witness.
Thank you. What is the normal time period for which vitrification is done for sperms in the IVF lab? Is there any standard time period?
Thank you for your question. Yes, scientists have not yet determined the exact period of time of sperm viability during cryoconservation. However, in medical practice, there have been several cases with storage more than 25 years. And today, the longest term for sperm freezing, which ended with healthy pregnancy, is 28 years. So modern protectants allow spermatozoa to maintain their functionally characteristic even after a long stay in cryostorage. Vitrification is absolutely safe method of raising male gametes. So keeping the material in cryobank does not absolutely affect the sperm. And through the entire storage period, the optimal condition for freezing maintained. So sperm can be stored as much as the clients need. There are no time limits. It's possible to store your own reproductive material in the clinic IVMED, which will provide opportunity to become a father in any age and regardless of circumstances. So that's all. Thank you.
Thank you. I had my own stimulation several times, but we received a few cells, and there were only 2 embryos. What would you advise? Maybe I should do IVF with cryocells? Question to doctor. Я переведу. У меня была собственная стимуляция несколько раз, но мы получили всего несколько клеток и только 2 эмбриона. Что вы посоветуете? Возможно, мне следует сделать ЭКО с замороженными клетками.
Действительно, что можно делать в такой ситуации? Особенности эмбриологии у человека таковы, что не каждый ооцит даст эмбрион, способный дать имплантацию, то есть дорастет до бластоцисты.
It's really an issue that not every human oocyte is able to provide the healthy development of the embryo up to the blastocyst stadium.
Чем меньше ооцитов получено на пункции, тем меньше вероятность, что на перенос будет хороший качественный эмбрион.
The less oocytes you receive as a result of a stimulation, the less is the probability that you will receive embryos for transfer.
Соответственно, повысить шансы для того, чтобы иметь большее количество эмбрионов, может большее количество яйцеклеток, полученных на пункции или пункциях. И если мы не можем получить большое количество ооцитов в одну пункцию, можно обсуждать накопление ооцитов, не оплодотворять их сразу одновременно, а несколько стимуляций, пункций позволит накопить достаточное количество.
which is why the more cells you receive in the IVF cycle, the higher are your chances to receive good embryos for the transfer. So in your case, if you cannot receive enough cells for creating the embryos, the good idea would be to freeze the cells that you receive and to run several IVF cycles and to accumulate the a higher amount of cells and then to fertilize them all together.
Когда будет накоплено достаточное количество яйцеклеток, одновременно можно их оплодотворить, откультивировать, и это даст больше шансов иметь большее количество качественных эмбрионов. Спасибо.
When you have the sufficient amount of the cells, then it is possible to fertilize them all together, to cultivate them up to the blastocyst stadium, and then you have a higher chance of having good embryos for the transfer. Thank you.
Thank you. Is there a time limit on how long embryos or gametes can be frozen for? Question to embryologists.
Yeah, I can answer this question. If you have your biomaterial in our IVMED laboratory, it doesn't matter it's sperm or eggs or embryos. It doesn't matter how long time it's kept in our cryostorage. It can be like 10 years, 20 years, it doesn't matter. In our clinic, we have like very modern cryostorage, and it can keep like as long as you want. And you can be sure that your biomaterial will be in safety and you can sleep well. Thank you.
Thank you. What's the sperm preparation technique for IVMED? Do you have some special technique for sperm preparation?
Thank you for your question. Of course, the choice of technique for semen preparation strictly depends on quality of the sperm. So if we have the sample with normal count, motility, and morphology, we usually use sperm washing or swim-up method. By contrast, with a bad quality sample, we usually prefer a density gradient centrifugation. We usually use for poor characteristic samples. It helps us to clean the semen after from the seminal plasm and the round cells, decrease the number of immotile sperm and get the great number of motile. So each technique can be changed and improved or improved with the sample changes. And also, I already talked about the special methods of preparation called as like microfluidic sperm sorting chip. I'm not going to repeat because you can see our record, but it's also a good method for preparation. Thank you.
Thank you. We would like to join IVMED guarantee program for surrogacy. My husband has noosomospermia, but 65% of sperm DNA fragmentation. Do we have chance to join this program? What is the best technique can help for the high sperm DNA fragmentation?
So for the question, of course, we are detecting a large amount, when we are detecting a large amount of sperm with high level of DNA fragmentation, we recommend to use sperm chip method. Sperm chip helps us to avoid damaged cells by different factors, for example, ROS, reactive oxygen species, and DNA fragmentation also. This method is based on the passage of sperm through special canals, and spermatozoa with the best quality will pass to finish line faster. And in this way, sperm chip allows us to select spermatozoa with the best progressive motility and with a normal level of DNA fragmentation for ICSI and without reactive oxygen species. So yes, we will have changes because we use the best methods, which helps you get good results. Thank you.
Thank you. Hello, I read that PGD24 tests come back as false negative because of unlucky cell selection for a test. Later, the embryos often correct themselves and turn out normal. How reliable is the PGD24 result? Can it be better to do PGD5? Is this case test for 5 will rule out the major abnormalities and give us gender? Amnio later will rule out the other problems. Embryologist maybe can say something.
Well, talking about PGT, it's always better to know more. Where basically a PGT was designed to avoid all the possible complications of the subsequent pregnancy that can be established after embryo transfer. That is why if we talk about information, it is always better to do PGT for 24 chromosomes for NGS. Because actually you can select the raw cells for PGT for 5 chromosomes and also for PGT for 24 chromosomes. And there is no really strong evidence that tell us that embryos are able to correct major chromosome abnormalities. So of course, as any other genetic test, there are risks of false negative or false positive result for PGT of any kind, but this risk is very low. It's like less than 2%, close to 1. That's why if you want to know more, it's better to do PGT24. But of course, if you are young and you don't have a very strong indications for genetic testing of embryos, you can go for PGT5 and actually screen your pregnancy very, very thoroughly. Thank you.
Can I add a little bit to this answer? Like if you're asking that the result was bad, and you probably think it was bad quality of cells that we take during the biopsy, we actually have very good communication with our genetic lab who perform NGS, like PGD24 and PGD5. So we actually always knew what problem was this. Is it the problem with cell quality or it just really bad result? So we can actually be really sure what result we give for you. So it's really important to have good communication with genetic and to understand what to propose for patient and like how to explain the results. Yes. Thank you.
Thank you. What is your criteria for donor egg freezing? If I choose donor egg from your bank, will I have high chance for good quality of embryos? The last question.
Okay, as I say, we have a really high survival rate about oocyte freezing. It doesn't matter quality of oocyte, but when we are working for donor egg bank, of course, we have strict criteria which we are choosing for the freezing and for cryobanking. Firstly, we have to analyze oocyte by assessment, high quality of morphology, and of course, we are checking a different stage of the morphology. We are checking quality of polar body, we are checking quality of cytoplasmic activity, also quality of perivitallin space. So till to freezing, oocyte is passing many different process, strict criteria for the vitrification process. I mean, our aim is not only to catch survival rate, but we want to create with this frozen oocyte high quality of embryos and of course in the first attempt pregnancy. So this is our main aim. That's why we are choosing under special criteria which we freeze oocyte. And soon we will add the advanced technology of the spindle weave. With spindle weave, we will have the chance also to get meiotic spindle maturity of the oocyte, and that will increase much more the chance of high quality of embryos after the thawing process. Thank you.
Thank you very much. And that was the last question for today. If you have more questions, please send us these questions to our clinic mail and we will answer them. Thank you, Birol. Thanks to embryological team and Dr. Veronica. See you next Friday on our webinar, which will be dedicated to problem of egg donation programs. And we will tell you more about program options at Medical Center IVMED. So see you next week. Stay healthy, stay safe. Bye.